Hello friends, welcome to lecture 3 of week 5. So in this lecture we will look at the concepts of assembly. We have been talking about what is assembly, also using the word hybrid assembly. So here we will look at what exactly we mean by hybrid assembly and what we do in this, and what is the contribution of short and long reads in this, what are the different algorithms that do this job, okay, and their advantages, disadvantages, right? So in this lecture we will focus on the various types of assemblies, the keywords that we use there, and the methods that cover those. So what is the concept of assembly? So basically it starts with the genome, okay. So there is a genome that you want to sequence, okay. It could be prokaryotic, eukaryotic, plant, animal, bird, any genome that contains basically chromosomes, either is bacteria or human, okay, circular versus linear chromosome. So like for example, this is a chromosome, right, which is actually DNA, compact DNA, okay. So you will be preparing library from this DNA, okay, and library will be, according to the sequencing method that you are using, or okay, that you want to use. It could be multiple methods also, okay, because ultimately you are dealing with hybrid assembly, so hybrid sequencing also has to be done by short read, long read sequencing, etc. Okay, along with the other steps that you do in the before proceeding for genome assembly, right, that we discussed in the previous classes, right. And then you will be having in hand files containing short reads and long reads if you have done both type of assemblies, right. So basically you started with a piece of DNA, you have broken that piece of DNA into small pieces, okay, small and long pieces, and sequence accordingly, and then trying to reconstruct the original piece back, okay. So basically this is the assembly process, and since there are many regions in the genome which look similar, identical, okay, that will interfere in the process. There could be heterozygosity, okay, like similar pieces but slightly differing in some base pairs, okay. There could be polyploidy, multiple copies of the same genome, whole genome or segmental duplications, right, paralogs. So these things will complicate the assembly process, and we will look at how the algorithms look into this, okay. Now again, one more thing, one approach is like you don't have a genome in hand and you're going for a de novo assembly, that means from scratch. So you have data and you are building the assembly from scratch, so you are trying to build the original chromosome or the genome, okay, from the reads, and you don't know which piece goes where, which piece goes to what chromosome, right, you have no idea, okay. And that is de novo, like from scratch. Another is reference based, where you already have a reference in hand, like in case of human genome you have a good reference now, right, and then you resequence using much lesser data, okay, and then you try to look for either identify some disease loci or some kind of changes that can tell you something about the disease health, or about that individual, right. So basically in the de novo assembled genome you will be having reads, you will be assembling those reads, long and short reads, okay, into contigs, okay. Contigs are like on the basis of overlap you will try connecting the pieces, right, and try to connect as much as possible till you don't find any other piece that can fit or attach to that contig, okay. And then based on some other information that you have, like insert size or longer insert size paired and reads, right, you try to build scaffolds where the contigs will be arranged in the right order, and in between there could be some regions which you do not know, and you fill that region as ends, right. Okay, so those are scaffolds. And then long reads and short reads together are used for fixing the gaps or correcting the errors, right. So you will see that in detail today, and ultimately you reach to the final genome assembly that you take for the genomic analysis. And here also like RNA-seq data is also important here, because that is coming from the exons, which are coding regions, and in any genomic analysis you would like to have more information and correct information of the coding regions. At least you like to reconstruct all the genes, okay, for the function analysis. So RNA-seq data acts as a empirical evidence, or supporting evidence to correct the coding regions, and also to make it more contiguous. Okay, so common terms that are used in sequencing and assembly process that we have also seen earlier, just a quick review. So basically contig, okay, so from the pieces of DNA sequences, you try to connect them based on the overlap, okay, and try to build a contig, okay, so that there is no more sequence which is available that can connect to that particular contig, okay. And then when you put those contig in the right order, like the way they would be arranged in the original sequence or the chromosome, so that is called a scaffold, okay. Though you may not have information about this part or this part in between, but at least you know that how they would be arranged in the longer sequence, right. So contig, scaffold, okay, and assembly is like as we mentioned, the process of putting pieces back, okay. Now draft sequence is when you are not able to complete the sequence, but there are so many gaps in between and many information may be missing, okay. So those is a draft sequence which could be like high quality or even low quality, or a working draft which you can use for further work and building the chromosome level assembly later, okay. Then if it is like plasmids, it will be dealing in kbs or like 1000 base pair. If you are dealing in a bacteria, you will be dealing in million base pairs genome sequences, MB. And in case of eukaryotes, it will be like few hundred MBs, like 50 MB like in case of fungi, or 500 MB in case of some plants and trees, up to several GBs or billion base pairs in case of large plant or basically more repetitive plants, okay. And mammals, it would be something like 2 to 3 GB, and in case of birds it will be like 1 to 1.5 GB. So all these genomes will have different genome sizes that you express in terms of like MBs or GBs. Then completely sequence you will call only when you have got the identity of each and every base pair and also their relative arrangement on the chromosome, okay. So that is a completely sequenced genome. And draft version is when some pieces are missing, and you can also have draft versions. For example, version 1 released in this year, then you do further improvement, have another version in the next year. Like in case of chimpanzee, I think this may be the case now because human is complete, so that also should also be nearing its completion, right. And other primates, there will be several drafts if you see over the years that are improving, and you can look at those drafts or the versions of those sequences. Then coverage is when you are like dealing with the sequence data, then how many times each piece or each region you have covered in your sequencing, right. So this part we have done in the previous class, so I will not go into details of this, but basically you will be going for a minimal level of coverage, so that either for short or long read, so that you can do it can achieve a good successful assembly and a good quality assembly also without gaps. That is very important, okay. Like typical example that I have given earlier also is the human genome. The first version came sometime in 2001. Then basically from that version it took almost 32 years to get a finished high quality complete human genome version, right, which was released last year. And so you can imagine that this process is not simple. A genome which is of interest to the whole world and scientists all over the world, okay, took so long. So that means it is really a challenging and process that involves a lot of basically complexities. Okay, so in the past we have looked at the various steps before proceeding for a genome assembly, right. For example, first of all you should know okay, so before these steps also there is one more step that I will cover in later classes. So that is the ethical consent. So before starting a genome, okay, either plant, animal, or any other organism, you should first take an ethical approval from your institute ethics committee or some authorized body which can provide you approval to do that study, because you will be dealing with in case of animal some tissues where you may be using some invasive methods, like you will be extracting blood or some tissue from the animal or plant or bird or any mammal, right. in case of plants, it may not be that that an issue, because you can take any vegetative part and use that for sequencing, right? Similarly, but still, like you should take approval before going for the study. So once you have identified the species and you know that this is the right species I am dealing with, you have extracted the DNA, confirmed the species by DNA sequencing by Sanger, then basically you have given the you've estimated how much data you want from that genome, right? Because you have some idea of the genome size from the plant C-value database, basically C-value database, or from like other researchers who have given some hints or regarding the number of chromosomes or the genome size in their previous studies, right? Or you can get an estimate like looking at what genome you are looking at and which family or which genus it belongs to, and if there are other genomes from that genus, that will give you an idea of what would be the genome size of your organism, right? So based on that you can get some rough idea, and based on that you can decide the coverage you want to go for long and short reads. Okay, and okay, so now you have got the data. Then the data that you get is not free of errors, and also may have adapters, primers, etc. So you will be doing quality filtering that we have already covered. So once you have clean quality filtered data above a particular Phred score, that you may decide. Okay, so basically you have done the adapter trimming, primer trimming, okay, removal of low quality reads, okay, and then you are left with the high quality reads that is needed for a good quality assembly, right? Then using that you can estimate the heterozygosity, because that will tell that how complex your genome assembly may be, because if it is more heterozygous, then it will interfere with the it will basically form parallel branches in the De-Bruijn graph, and that will also interfere, right? And similarly, like 1% or more heterozygosity is generally not good, right, for assembly purpose. And ploidy, because if you have multiple copies of the DNA of the same chromosomes, okay, generally humans are diploid and triploid or tetraploid, so that will also interfere in the, because they then haplotype resolving will become an issue. Okay, then estimation of genome size you can do by k-mers that you get from your short reads, right? That will give you some estimate of what the genome size could be, or you have some literature or other evidence of genome size that is important for going for the genome assembly. Okay, so basically I will give you some illustration of what you are going to deal with. So basically from the cell you extracted the chromosome, DNA, and DNA is basically gene, non-genes, non-coding RNA, and those repetitive sequences and other regulatory elements in the genome. So anything can come in your DNA fragment. Okay, so basically you are kind of reading a book, or you have a book and you want to know that what is it contains. Okay, but the problem is that you cannot you don't have the assembled chapters and you cannot read it like that. So you can read it line by line, and the lines you get is like you take out all the pages from that book, okay, and pass it through a paper shredder such that the paper shredder cuts only one line, okay, and mixes all those lines together, right? That is what a sequencer also does to your genome. Okay, so basically because sequencer cannot sequence long reads or two long reads or the entire genome, so basically you will be dealing with pieces of sequences, right? Okay, so now in this case you are having lines only, okay, and you want to construct paragraphs from those lines and paragraphs into pages, okay, and pages into chapters and chapters into the entire book, okay, and the relative order of pages also, right? So that complex problem we are dealing with, okay, and that is why the long and short read data, their features that are important for the assembly, we will be using all those features. Okay, so one example, like for example there's a line here, it was the best of times, it was the worst of times, it was the age of wisdom, it was the age of foolishness. So you can see that the words look repetitive, okay, and it's a single sentence, right, from Charles Dickens' A Tale of Two Cities, right, and you can copy it 5 times, okay, and cut it in 5 words, right? Then you can, those 5 words are like, you know, kmers, right? And now you want to construct the entire sentence, okay? So basically like this, you have made it 5 copies and like 5 words in overlapping fashion. If I derive from this, like overlapping keywords from this text, okay, so there will be some portions which will be repeating, because you can see it was the off times, it was the off times, it was the, okay, it has coming 2 times, right? And right, and then you have to build, you know, connect those keywords into the way that you can have the shortest path, or you can say that you can connect all those pieces, you can walk through all those pieces, right, and construct the entire line, right, without getting confused with the lines that are not basically completely identical, or they can fit anywhere, because there could be several genomic regions that may fit anywhere, and there will be several repetitive regions also that can come from any part of the DNA, and you don't know where to fit it, because it's just a sequence that contains only repeat, okay. So and you will be dealing with like billions of kmers when you are dealing with the genomes, because you will be forming nodes and edges, and basically that will be almost in the proportion of your genome size, okay. So there are 2 main genome assembly algorithms, De-Bruijn graph and overlap layout consensus, OLC, and DBG, that you call them in short forms, DBG and OLC. And okay, so we'll be dealing with these algorithms today. So basically De-Bruijn graph or DBG is to assemble the short reads, okay. So it's a like a mathematical way of constructing the sequence, where basically each node in a graph that you will be forming, okay, will be a kmer. So you have derived kmer from your DNA. So basically this was your read. So I'll be reading that into overlapping kmers, right, okay, with a certain window size that is actually equivalent to the length of kmer, right, and of length k. And basically you will be deriving kmers for this size, like equivalent to the length of the DNA minus 1, okay. You will be deriving that many kmers from one read, okay, and then you will be constructing or arranging those kmers in the form that you can read them or connect them together. So you will be seeing here, okay. So basically in DBG, as I mentioned, that you are trying to connect 2 nodes which are neighbors in the genome, or 2 kmers which are coming from the same part of the genome, okay, and the node number here will be equal to the genome size, okay, okay, g minus k minus okay, plus 1. And also basically it links, and the link number is also equivalent to the genome size, that how many times you are linking it, and it is unrelated to the genome sequencing depth. So even if you have generated like massive amount of data, okay, it will not increase the number of nodes in your graph, although initially you may need a lot of RAM and computing, but later on the assembly is not difficult, okay. So like you are reading a read and dividing that into kmers of say length 3, okay, right, okay. So now from these 3 reads which are okay, you have first of all you are making the kmer, then you are building the graph. That means the kmer that you have derived, like TTC, TCT, TCTA, CG, okay, you are connecting them in the order, right, and basically each of the kmer become vertices in the DBG, okay, and edges connecting the overlapping kmer. So those kmers which are overlapping are connecting together by edges, okay, and there will be situations where there could be polymorphism or heterozygosity, and that will be forming some parallel branches in your graph, okay. And then you will be walking the graph and connecting the kmers which are overlapping, and that should be connected together, okay. And then basically CGA, GAT, right, AT, ATA, ATT, like that. So basically a sequence of CGA, T, and like that. If you connect this together, you will get this sequence like CGA, then GA was also common here, T, CGA-T, CGA-TT, and like that you can connect the k-mers together. Okay, so this is De-Bruijn graph that is used for short reads assembly, and the advantage is that basically you don't actually need to worry about the paired end reads, or information about like where the reads are coming from, and right. But basically the problem with this that you will face, that it could be that because you have done over breaking of the read. So okay, so length of k-mer that we have mentioned about earlier also matters here, because there is no fixed rule that what length k-mer you should pick. So basically you should increase by 10 or 11 lengths. For example, you are dealing with a read length of 150 base pair, so generally like k-mer size could be half of that read plus 10 or 11. So basically it could be like, if the read is like 75, you can take like 85 or 81 or 91, and like that you can. So basically multiple k-mer lengths you have to try, okay, to reach. So you can do this exercise on a smaller set, okay, and then find the best k-mer and do the assembly process, right. So this length of k-mer, and also like too much breaking of the reads, and there could be pieces that may look exactly identical. Okay, if you take too long piece, right, too long a piece, that will also increase the number of nodes, and if you take too short a piece, it will like match multiple places in the genome. So that could be one of the issues with the De-Bruijn graph. And there are so many short read assemblers which are available based on this approach, of which like Spades is the most common, okay, released. The latest version came last year, and okay, and number of citation, like number of times people have used it in their work, or basically reported published work, it is like more than 18,000, and yearly more than 1800 people are reporting it. Okay, so this is the most widely used short read assembler based on this approach. Okay, then Velvet is there, Soap de Novo, MetaHit, Abyss, IDBA, All Paths, Masuka, Ray, SGA, so many are there, and many are coming, and basically these are old but not much used, okay, because the algorithm that are better performing and robust, and also not very much RAM and memory intensive, okay, and which like easy to use, easy to install, those are more popular, and that is why like Spades, Velvet, Soap de Novo are one of the popular ones. So in the case of long read assembly, we follow the approach of overlap, layout, and consensus, right. So basically since the reads are already very long, so k-mers, deriving k-mers may not be a good idea, and you can look at a decent overlap between the reads. Okay, so that means there will be all to all blast or something, pairwise alignment using some of the available methods, and then the pieces that are showing a consensus, or basically overlap, a reliable overlap, okay, with above a particular threshold will be taken together, right, and then those reads which are showing the overlap are laid in such a way that based on their overlap that we have observed, okay, that is the overlap layout, and then based on that layout we will be building the consensus. Okay, so like here read 1, read 2, read 3, okay, so like read 1 and read 2 are showing some overlap, read 2, read 3, and read 1, read 3. So you will be arranging them in the order of their overlap, and if these are the three reads, read 1, read 2, read 3, based on that you can build the consensus. So overlap that you choose because you are dealing with reads which are like several kbs in length, okay, even hundreds of kbs in length. So overlap of 1, 2, 3 or 5 kb is a routine practice, and this also ensures that the two reads that you are considering are really aligning, or basically can get connected together, right. So basically in OLC, overlaps are computed by comparing all to all reads, as I mentioned, using some of the alignment approach, okay, and those which are showing a good overlap are connected together and form the contigs, okay, and basically those contigs are then extended further to build the highly contiguous sequence, right. So overlap that we have so overlap layout consensus is, that is why this method is called, because it is looking at the overlap, then arranging the reads according to the overlap and building the consensus, right. So here the node number will be equaling to the number of read number, because if you increase the reads, or if you have dealing with a lot of data, so that many number of nodes will be there, and that many number of all to all blast you will be doing, or any alignment you will be doing like MAMA etc., right. And that will really put burden on your memory, RAM, and basically your CPU. Okay, so and that is why dealing with the long read assembly may be challenging as compared to the short read assembly. Okay, so basically for the genome assembler for long and short read, if you see, so you will be starting, just to like again giving an overview of the thing, you will be starting with long with the genome in hand, producing long and short reads by either of the methods like Illumina or 10x, right, link reads, or the low quality long reads by PacBio and Nanopore. Okay, now these all are also becoming better and better, but still like they are much lower quality compared to the high quality reads. Then basically for different type of reads, you will be using different types of assembler. Okay, that will be dealing in today and in the later classes. And basically these assemblers like Kanu, Falcon, Fly, these are long reads are based on OLC, short reads are based on generally DB graph, DBG. Okay, and that is how these two algorithms that we have discussed, OLC and DBG, are common in the methods that we will be going to use in the assembly, although the methods will differ in some of these steps, or in their basic way they are doing the assembly, but overall like they will be using the same type of algorithm. Okay, so we have already discussed this, that we will be starting with DNA, producing long short reads, and then once you have done the hybrid assembly, then you will be using, based on the statistics that you have got in the hybrid assembly, okay, you will be going for the chromosome level construction using Hi-C or another method. Okay, and that will actually improve the quality of your assembly and also produce a more contiguous DNA fragment which is equivalent to the chromosomes. Okay, so just to give you an idea of, like, if you are taking a genome of like 1 GB or 1.5 GB, so basically the first few steps, the high quality and the short and long read assembly may take 7 days. Okay, depending upon your system. So basically assuming here you have a system which is having a RAM of 500 MB and basically your 500 GB, right, RAM is there, and your number of cores are 64 and above. So basically this much time you will take. For the scaffolding and merging step, it may take 2 days or longer. Okay, but for the polishing steps, where you are correcting the errors and using the long and short reads together, basically where you are constructing the hybrid assembly, okay, that will be taking a lot of time and more than 2 weeks during that time. So basically overall the assembly process, if you have decent resources, can be from start to end it may take a month or lesser, right, for most of the genomes. And then you're determining the genome statistics like N50, BOOSCO, LAI etc., or basically your gene prediction, and those will take more time later. Okay, so there are several methods which are people have published papers that give overview of how to deal with a new genome, or the steps that you may be using for analyzing a genome, the tools that you will be using, okay, and how to install them, and also taking a example and running through it, and looking at how that example you are using the multiple steps of assembly for that example. So one tool is here in start protocols in this paper. So basically this you can go through if you want to see the these steps in a single paper. right, which basically starts with like genome size estimation, read raw read statistics that we have done, de novo assembly, and then quality assessment of the assembly, N50 and LAI or BOOSCO, etc. Then scaffolding and building the complete chromosome, and then calling for structural variants or duplications or the evolutionary analysis, including science of adaptation, points of selection, etc. Right. So typically for the first few steps like genome size estimation, this it may take about 2.5 hours. For basically this is example for the Drosophila genome. Then for assembling the contigs, about 2 days. For quality assessment, BOOSCO, coverage, jellyfish, etc., those steps like about 30 minutes or more. Okay. And for the assembly and variant calling, like few hours. Okay. So this is like a rough idea for a smaller genome. Okay, then this gives you some idea of how much CPU and RAM it may take, and given a particular amount of data. For example, you are dealing with the E. coli genome, which is something like 4.6 MB, right? In length, and you have generated like 50x data of this. So 50x of, if I consider it 5, so 250 GB data, 250 MB data you have using. So it will take about 2 hours and 2 GB RAM on a system which is like right. So and similarly, if you go further for human genome, if you see, and for Drosophila, it will be taking a huge amount of RAM, 75 GB to 714 GB, and then also the CPU time also increases, right? If you have more number of CPU, the CPU time will decrease. It is per CPU time, right? So parallel computing or HPC helps here. Okay, and typically the rule is that you should have at least 10 times the RAM of the data that you are dealing with, because although the algorithms are becoming better and better, but still they are memory and CPU intensive. So if you are dealing with say like 250 GB data, then you should have at least 10 times more RAM to do that in time. Otherwise it will take a lot of time. To your process may run, but it will be like months long analysis time. Okay, so basically in the hybrid assembly, it is not about mixing the long and short reads together to just build the assembly. It is there are algorithms we have seen, like if you want to assemble both long and short reads separately, but in the hybrid assembly you will be using the features or the significance or basically attributes of long and short reads, which can help to build the correct and contiguous assembly. So we will be looking at those algorithms. Okay, and why we need to do a hybrid assembly approach using long and short is because the genome that you are dealing with may be a polyploid genome, okay, having multiple copies that may lead to problems in haplotype resolving, or it could be highly heterozygous genome that will interfere with the assembly process because of the mismatches you will see in the fragments during the assembly. And then it could also be like too big a genome, the genome size, or it could be highly repetitive genome that will also interfere with the process of assembly. And that is why we need both long and short reads, because that way we can, like repeats we can resolve by longer reads, and heterozygosity, and this we can get a better idea of what base pair should go into the consensus sequence by the shorter reads. Okay, so basically in the hybrid assembly we will be using information from both second and third generation sequencing technologies. So either using the third generation to support the second generation sequencing technology assembly, okay, TGST or SGST that we are calling here, okay, short second generation sequencing technology and third generation sequencing technology, and then basically using either of those as the template and using the other data from the other technology to correct it or to make it more contiguous, right? So because like long, as I mentioned, long regions can be covered by long repeat regions can be covered by longer reads, accuracy by short reads, and combining together will give you correct as well as contiguous assembly. Okay. So basically, there are several tools which are available that can do the hybrid assembly, right? We'll be dealing with two important tools today. Okay. And so either in this case there are three ways you can do the assembly. One is the assembly of long reads can be corrected using the short reads. So it could be that you have long read you have assembled, and now this is one of the contig that you have got, but there could be errors at multiple places. Okay. So when you are using the short read data, right, then you can map this data here, and you can find out, okay, what is the base pair in the short read, okay, long read contig and short read. So you can check the same base pair, what is the correct base pair at that position in the short reads, and correct this error, and this way you can correct the long reads assembly. Or you can use the short reads assembly as a template and try to fill up the gap by using the long read assembly, right? So that is why there could be type 2 or type 3 type of merging. Like type 3 is quick merge, that uses the one of the assembly as a template, another one as the basically as the main assembly, another one as the reference assembly, and this way it tries to merge both the assemblies together. And some of the pipelines are also available using that provides short and long read, can take both of them, right, and can do the hybrid assembly. Okay. So either you can go for, so you have short read data and long read data. Okay, short read and long read data, you can assemble them separately. Okay, get some contig level assembly, right, and then use either this one to improve this one, or either use long read to improve the short one. Okay. So basically you have done the short read assembly, de novo assembly, and assembly scaffolding is done, and then at this step you can use the long read data to increase the contiguity, right? Or you have done the long read assembly, de novo and polishing correction, and then at this step you can use the short read data for scaffolding and also the polishing, right? And then you can merge, and again you can go for another round of gap closing and polishing using either long or short read. Okay, so in this way you can use both the technologies, long and short reads, to achieve the better quality genome assembly. So this is typically a type 3 hybrid assembly, which is like used by quick merge and RagTag, which we will be seeing in some detail today. Okay, so quick merge is a hybrid genome assembler, okay, which takes two FASTA format file, okay, that is like the long and short reads, okay, and basically it tries to merge both the sequences together. So if and basically first of all it tries to find the alignment between the sequences, okay, so using the MUMMER program, that is okay, before merging the assembly. So this is, I will come back to that. This is the paper that you may like to read before going to use quick merge, because that will tell you how to use it, and how it was developed, and other important information about the tool, and you can download it from here. Now if you look at this tool, basically what it does is it uses the so you can have either PacBio based or ONT based assembly here, okay, or you can have the short read assembled here like De-Bruijn graph, or like this is OLC based assembly, this is DBG based assembly, long read and short read. So you can have two assemblies, and then it uses a tool called MUMMER, which is a commonly used tool to do quick alignment, okay, to identify the high confidence overlap between the like read the contigs from either of the assembly. For example, here it will see that this is a blue line which is coming from here, okay, and this is a orange line which is coming from, it is trying to find an overlap between the two, okay, and then this line is coming from here, right? So it will find the regions which are high confidence overlap. So you can see that this is a very small overlap, this is a reasonably good overlap, this is a reasonably good overlap, right? So it will identify the high confidence overlap using the Mummer tool, right? And then based on that overlap which it has found, those high confidence overlap, it will use that, those. So basically that tells that those are the contigs from either long or short read assembly, right, that must be connected together. Okay, so basically those and which can be connected together, and those contigs are now connected together and form the seed. Okay, so basically it forms the seed contig from those. So if you can see here that in these cases the overlap is good, right, whereas in this case the overlap is poor. So HCO score is too low, okay, HCO too weak, okay, whereas in this case you can see the strong line or the dark line, and this is the dotted line or dashed line, which tells that this should not be connected and this should be connected together. And you can see here that, and those which can be connected together have been connected in the form of merge those contigs based on the overlap, and then you are building the consensus using both the information from the long read and the short read assembly. Okay, so basically quick merge identifies seed contigs in a cluster above a certain size and threshold. Okay, so size also matters here. They should be having a long as well as short contigs from long and short read assembly, but above a particular threshold. It is not that all the small and unimportant or basically not so significant aligned regions, okay, or poor HCO regions will be connected together. Okay, and then it will try to connect all those to build the complete genome. So basically there is a donor genome and acceptor genome concept here. So that means you can use either of the assemblies, the nanopore or basically the long read assembly or the short read assembly as the template. Okay, so basically this was the thing. So Mummer is generally used to identify the high confidence overlap regions, and those are the contigs that shows high confidence overlap and they should be connected together. So those are connected from the long and short read assemblies, right? And then once the graph connecting those contigs has been constructed, the contigs in the graph are spliced together with the donor genome's content preferred over the acceptor genome. Okay, so that means you will be giving importance to either long or short read assembly in this case. So generally like the donor in this case should be the short read assembly, because that will be more accurate and that can be used to fix or basically fill up the gaps in the long read assembly, and also to improve the errors which could be there in the long read assembly. Okay, now the another tool, hybrid genome assembler, is RagTag, which is basically also merges the assembly, but it has some different steps. So basically the idea is to again merge the long read and short read information or the assemblies together, right, into a consensus sequence by including regions from either of the assembly. Okay, so basically it does three things. One is does the scaffolding, patching and merging. So in the scaffolding you can see that okay, and one more thing which is correct. That means there could be some misassemblies and it would like to correct. So it has one step related to that. Then in the scaffold you can see that in the query there was some missing information that was quoted as ends in those scaffolds, because scaffolds are something where contigs are joined together in possibly correct order, and in between information, even if it is not available, is filled up by ends. Okay, so basically they are connected together with reference to the reference. Okay, and then patch is using the in patch tool, basically using the pairwise whole genome sequence homology. It tries to join the two contigs without introducing the gaps. So basically it will fill up the gaps in a target genome using information from the query genome. So basically in the target genome, if these were the gaps like ends here, right, so it will be filling up those gaps using the information from the query genome, right. So basically target and query is nothing but two types of assemblies that you are dealing with, the long read and short read assembly. You can consider use either of those as target or the query. Okay, and then third thing is like you can merge two or more distinct scaffolds to form the genome assembly. So using the information from the target and query or long and shorted together, it will merge them together and form the contiguous assembly. Okay, so just to give you an idea of the patch thing again. So basically in this target, basically this pieces were missing. Okay, so right, for which you do not have the information. So from the query, which could be your, so target could be like smaller contigs that you have got or scaffolds you have got from the shorted assembly, which are more correct, but the longer query, that means this query here could be the long read contigs, right? Those regions are used to fill up these gaps and building the more contiguous assembly, right? So this is way in which the RagTag works and does the hybrid genome assembly. And you can see that in case of quick merge or RagTag, basically information from both long read and short read assemblies are used together in such a way that you can use the correctness of short read and contiguity of long reads to make a better consensus and reliable, accurate and contiguous assembly. So you can read more about the RagTag from this paper, and this is available at their server, right? And just to give you an idea of like again, okay, a kind of quick revision. So basically you will be starting with short read and long read data, then you will be doing short read assembly by any of the DBG based or De-Bruijn graph based assembler, and similarly long read assembly by any of the OLC based assembler, okay, like Kanu or Platanus here. Okay, then you will be performing the hybrid assembly using either of the hybrid assembly tools which I have mentioned, or another tools which are available, right? And then at various steps you will be doing the polishing. Polishing means that now a consensus is built using the two assemblies, merging the two together, but still you can correct the final assembly using either information from the shorter reads to fix up the errors in the assembly, or from the longer reads to fix up the errors in the assembly. And then once you have got the final assembly, also using quick merge or some other tool. So basically for the polishing, pylon is one of the tool that is very commonly used and which will also be dealing in the next class, right, in next lecture, and then followed by quick merge and further polishing steps again to further improve the assembly process. Okay, so basically your assembly process is a multi-step process that includes sample collection, sequencing, assembly, polishing, correction, scaffolding, merging the assemblies together, building the consensus, going for the Hi-C or chromosome level construction using Hi-C, and then doing the genome annotation by gene predictions, non-coding, coding part prediction, function analysis, right, and also using the information from the RNA-seq data or the transcriptome data to improve the assembly and also improve the gene prediction and the function annotation. Okay, so in this lecture we looked at the various concepts of genome assembly, what is a genome assembly and what are the terms associated with that, what is hybrid assembly, what are the different approaches that are used for short and long read assemblies, okay, like De-Bruijn graph and OLC method, and what are the tools that are based on those methods and their significance, and also how the tools that merge the two assemblies work and what is the criteria and concepts in that and what is the outcome of that, right? So this is good for this class, and we'll be meeting again for the next lecture. Thank you.